Imagine if you could look at a random splash of seawater filled with tiny extraterrestrial creatures. We could find something as wonderful as this in these plumes from Saturn's moon Enceladus. A chance for a super positive outcome. Cassini has sampled the plumes already, flying through them. This has no chance of contaminating its oceans, a gold star for planetary protection, so this is something we know we can do. Enceladus, on the left of this slide, is a sure thing as its plumes erupt continuously, feeding Saturn's E-ring. Europa's plumes are much more uncertain. They could be short-lived, like this Icelandic geyser. The 200-kilometre high plume that Hubble saw, shown in blue on the right, could have been made by large impact on Europa, so it's not yet proof of geysers. When Cassini passed Europa on its way to Saturn, it saw nothing. He would have been able to spot plumes extending far into space, easily. So I'll focus on Enceladus for now, as its plumes are a sure thing already. From time to time I'll mention Europa as well. Enceladus's ocean is kept liquid by eccentricity tides. It is kept in an eccentric orbit by resonances with Dione, and so, with a steady spin rate, it can't keep the same point on its surface facing Saturn, and its surface crust gets tugged back and forth by Saturn's gravity. It loses heat constantly through these tiger stripes, and the heat budget is a long-standing puzzle. It's hard for eccentricity tides to generate this much heat. Some models work, but they have to make assumptions about its entire interior. There is geological evidence which suggests that it is freezing slowly. We don't need to worry about the next few thousand years, but the geysers may stop within a few 100 million years. The safer evidence suggests that its ocean is at least 1 billion years old. There are many different ideas currently on the nature of Enceladus' ocean. Its interior is probably like a carbonaceous chondrite meteorite, unprocessed early solar system material, rich in organics. That's great for life, except that it's also expected to be completely undifferentiated and porous, which is not such great news for life. It's thought that all the salt from its interior has already flowed into its ocean. So one possibility is that it may have reached chemical equilibrium, and any life run out of food, extinct or sparse. However, tidal effects might easily supply enough energy for a complex ecosystem. Cassini's measurements suggest two main possibilities: a very alkaline soda lake, or less alkaline white smoker type ocean with hydrothermal vents with hot rocks. So it could support complex life like Earth. On the other hand, its ocean might be young, only 1 billion years old, or it might be that life evolved there more slowly. If so, perhaps it has only early stages of life, or no life. There's a tendency to look at this and say the right-hand side is what astrobiology is all about, complex life. And for the left-hand side, with only early life or non-life, let's leave it to the planetary geologists. We might even feel a duty to bring Earth life to a lifeless ocean. I'm going to suggest that early life or non-life is also of extraordinary interest for astrobiology, and as well, we have nowhere near the understanding needed as accidental or intentional ocean eco-engineers. This visualization shows how Earth life makes proteins. Here, messenger RNA produced by DNA polymerase meets a ribosome, a huge complex molecular machine that spontaneously assembles around it. In this visualization, those green triangular transfer molecules each bring a single amino acid to the emerging protein chain. You can see the amino acids as red dots at the tips. This is going on at this speed in every cell of every known Earth creature, and this visualization just hints at the complexity of the processes. It works, but it seems Heath Robinson, or Rube Goldberg if you're from the States, like the professor's potato peeling machine. Life must have started simpler. Perhaps a self-replicating polymer, not yet alive, but encoding information. Or perhaps an autopoietic cell. This means it takes in ingredients uses them to recreate its metabolism and cell wall, and expels wastes. It's made up of only a dozen or so chemicals, and it just splits when it gets large enough, or a daughter cell from inside escapes. This graph shows time of origin against gene complexity on a log plot. Prokaryotes, cells without a nucleus, are oldest. Continue the graph back as a straight line and it crosses the axis at around double the age of the solar system. So, perhaps life started on an exoplanet around an older star. This passes through a nebula where our solar system is forming at the tip of one of these fingers. It misses most of the embryo stars, but by chance comes close enough to ours to transfer life by meteorite impacts. It can do this from quite a distance. In this photograph of H.L. Tauri, planets could be forming in one of these dark bands. Or, the solid line may just show the characteristic slope for modern DNA with error correction. Perhaps earlier life evolved much more quickly and in our solar system. Life in these extraterrestrial oceans could be anywhere on this graph. This is a good point to give a quick mention of Europa, which, with its oxygen-rich ocean, could even have the likes of fish and octopuses. It's unlikely that after billions of years, Europan and Earth life reached exactly the same stage of evolution. Modern multicellular life is quite recent on Earth, so it might not have evolved there yet. Or it might have evolved to some future form of life that we don't have yet. Could Europa have forms of life as far advanced beyond us as we are advanced beyond the first complex multicellular creatures? Perhaps it's 50-50 either way if asked to venture a guess. And if we got that far, what about a civilization, millions of years old? In an ocean with no fire, their technology would be limited. They would know nothing about us, and we would know nothing about them yet. While in the other direction, if they are less evolved than us, well, more than half of evolution is hidden from us, according to this graph of complexity. Each pixel here covers over 70 million years. So our lab experiments like Yuri Miller, simulating conditions on early Earth in a flask, explore a tiny fragment of the first pixel. That leaves a huge gap, which must have steps in it as momentous as the nucleus of eukaryotes, multicellular life, plants and animals. But what those are down here, we can only guess. In some ways, that might be the most interesting of all, to fill in a little of this gap. That's why I suggested an early life or non-life ocean could be just as interesting for astrobiology. Then, life in those oceans, whatever their stage of evolution, could also be either related or unrelated to Earth life. If life evolved around another star and the oceans are ancient, we could be related. But if the Europan ocean is young, and any previous life long decayed when it formed, or if life evolved in our own solar system and never got transferred to Europa, it is likely to be independently evolved. There's still some possibility of a common ancestor, but with perhaps at most 10 meteorites exchanged over the entire history of the solar system, chances are limited. If independently evolved, would it still resemble Earth life? Earth life may seem optimal. It stopped at 20 main amino acids, after all, and that's with a lot of spare coding capacity. Here we see that serine, for instance, is coded in 4 different ways. Each of these could have coded for a different amino acid. However, this could easily be a local optimum. Let's think of height as anything you want to optimize, such as efficiency of metabolism of the entire cell. You start in Australia and keep walking up, and eventually, probably, you just get to the top of a small hillock. If you allow yourself to go up and down and travel great distances, the highest peak you reach, Mount Kosciuszko, is only a little over 2,000 meters high, less than half the height of Mont Blanc. If you're at the very top of this peak, because it's a summit in this analogy, any tweaks, such as adding more amino acids, always takes you downhill, so you might well think Earth life is the best that any life could possibly be. But if you started in North America, you'd end up on the top of Mount McKinley, which again would be a summit and seem totally optimal. Everything you try takes you downhill, unless you were able to explore as far as South America, and then both of those would be a lot lower than Mount Everest, which you reach as you gradually explored Eurasia. So there may be many different optimal best solutions for life, and we only know of one which is used in all Earth life, even the oldest cells, the prokaryotes. So life on Enceladus could be better than us, the Mount Everest of metabolisms. Or Earth metabolisms could be the Mount Everest, and Enceladus life is relatively feeble. If Enceladus does have life at all, it is perhaps 50-50 whether it is a more optimal solution than Earth life, or vice versa. Specialists who study the interior of a cell sometimes compare the complexity of the interactions to an ecosystem. In this analogy, all Earth life has the same ecosystem inside. I've listed a few of the things in it. Experts could add many more, common to all Earth life. This ecosystem is so similar, life forms often swap genes in parallel transfer, and they still work. Here's a gene from a fungus, which gave this pea aphid the ability to generate carotenoids, which turns it red like a carrot. Imagine if the African savanna was the only ecosystem biologists knew about, with minor variations. Then one day, we discover a coral reef. We have never seen a tropical rainforest, tundra, a seashore, or anything. This shows how much we could learn just from observations of a single cell or protocell based on different principles. So of course we are careful. This is one of the Viking landers, gold standard of planetary protection on Mars. It had at most 30 cultivable spores over its entire body, and so probably not much more than 3,000 spores or dormant microbes in total on launch from Earth, and probably far fewer when it reached Mars. Nevertheless, you can question if it was sterilized enough. The aim was to have no more than a 1 in 1,000 chance of contaminating Mars during the exploration stage, which is how they were thinking about it then. Is that enough, giving them the chance of a super positive outcome? Anyway, that decision is made, and it's generally thought we have not contaminated Mars with Earth life, mainly because conditions there are so harsh. But it is important to know we haven't yet looked at our spacecraft on Mars to see how many dormant microbes they have, and in what species. Our calculations predict that there are microbes, and that they are sitting there doing nothing. Again, we don't know for sure, because we haven't checked. For instance, Phoenix was crushed, and one of its solar panels snapped off with the advancing winter dry ice sheet meters thick. We have no experimental data from Mars on this, on what happened to the microbes on it. So this is a little bit shaky. We don't think we have contaminated Mars, but it's not the best basis for making decisions about icy moons. The National Research Council looked at it, and especially since these oceans are much more habitable than Mars, and probability assessments are more uncertain here, this approach just wouldn't work. They recommended binary decision trees with a yes or no answer at each stage, rather than combining probability assessments. The orbiter or flyby passes the test at stage 5 with a yes, because it's not going to encounter a habitable region. But any Enceladus ice mole or penetrator or cryobot passes all the way through to the final stage. Then you have stringent planetary protection or cancel mission. The animation shows one idea for how a future ice mole could explore the Europa Ocean. The Russians in Antarctica drilled nearly all the way down to Lake Vostok and then discovered they were approaching a lake. So they just stopped for over a decade. Eventually, they drilled into it in such a way as to trigger a mini geyser of 30 or 40 meters, so that there would be no form of contamination into the lake. This is like a flyby mission. So far, they have only sampled this plume. It would be so easy to send a submarine down, but they feel they haven't yet got the technology to sterilize it. So in the same way, when exploring Enceladus, we should always have the option, cancel the mission, not ready yet. This rabbit-foot fence was built to protect Western Australia in the 1900s from rabbits introduced by European colonists. It was the longest fence in the world, and it did work after fashion for some years. Cartoonists had a field day, showing here probable use that rabbits might make of it. I wonder what kind of a fence we could put across the Enceladus Ocean. COSPAR recommends a target of a chance of 1 in 10,000 of contamination in the next 1,000 years. However, there was a lot of discussion. There's no science you can do, no a priori calculation, no experiments that can give you these two numbers. It's an ethical decision. If you think about it, it has a potential super-positive outcome. You might look at these numbers a bit differently. We have a chance of a super-positive benefit to billions of people, revolutionising, perhaps, medicine, agriculture, nanotechnology. We have no idea how probable it is, but it is a possibility. In the direction of sample return to Earth, the legal situation is clear. The precautionary principle applies, a principle of modern international law, introduced because of the new things we can do. A sample return could, in the worst case, impact on habitability of the Earth. For instance, if we return a life form better at living here than Earth life, it could degrade the environment. In a situation like this, we must take precautions, even if we don't know what's in it. It's no good saying, "I've no idea, but surely it's okay," or use hand-waving arguments. We have to take precautions anyway. And it's us, the proponents, that have to do the science, and the discussion has to involve all affected parties, which in this case may mean the entire world. We can never say, "We are the scientists, we will make the decision for you." That's totally wrong. And you have to consider all possibilities, including always no action. That can get forgotten. If you ask experts, "How can I return this sample safely?" that sounds innocuous enough, but that's the wrong question. You must always have the option to say, "It is not safe yet," if that is what their research shows. I suggest it should be the same for super-positive outcomes in the forward direction. We should take precautions even though we have no idea what is there. As proponents, it is up to us to prove that what we are doing is not going to impact on it. And I would say, because this is super-positive, it impacts on the entire Earth, because of those billions that would be affected in a positive